Hi, I'm Jyoti Devakumar, and I'm co-chairing these sessions with Bernie Siegel. And I'm given to understand that I have 3 tasks to do today and for the next couple days. One, to make sure to invite and introduce the speakers. Two, to make sure that they stay within the allocated time, which I enjoy doing. And the third thing is to moderate all the panel discussions that happen and to also make sure there is audience participation in asking questions to the speakers. And the fourth thing I've assigned myself is to make sure that I have fun in all these, and all of you should also have fun. So our next speaker is one of my favorite speakers, Andrei Gudkov, and he is a professor of oncology and also the chair of Department of Cell Stress Biology at the Roosevelt Park Cancer Center. And he is also an entrepreneur. He has a company which fixes a SINE and line element damages that occur in the cell with age, and it's a retrobiome company. And he's one of the powerful speakers I know. If most of you in the audience, or if some of you in the audience do not know what SINE and line elements are, don't worry, he's going to educate you. He is one of the textbooks as far as the SINE and line elements are concerned. Thank you. Andrei.
Good afternoon. Thank you for the invitation, and Jyoti, thank you for a nice introduction. I will talk with you about something which most people don't realize, although I think it's becoming a progressively more and more important topic, both in oncology, in aging, and in other areas of biomedical research. So the work which I'll be presenting, it has a number of references to published work, but mostly unpublished, but will appear soon because papers have been submitted already. It's a group of a large number of my colleagues from Roosevelt Park Cancer Institute and from the company named Genome Protection. So again, I will be talking about aging, and the exciting and provoking puzzle in aging is that during last 150 years, we managed to gain lots of extra years in average lifespan, but we gain nothing in the maximum lifespan, indicating that age-related diseases, which we so effectively learned how to treat, is not equal to aging, meaning that aging is something which is ticking in us regardless of age-related diseases, which can only be accelerated by aging. And from many, many different combinations of thoughts about aging, there are 3 which I think majority of people agree on, is that A, in mammals, aging is genetically determined because we have a very genetically programmed longevity in every mammalian species. It is associated with accumulation of cells with damaged DNA and also associated with acquisition of chronic systemic inflammation. So I will tell you 2 stories today. One of the stories is about whether accumulation of DNA damage is indeed enough to initiate aging and provoke aging. And the second one will be about what is the source of generator of aging, of DNA damage inside our organism. So story number 1 starts from our naive idea to accelerate aging and to save money waiting until mice become 2.5 years old and start developing age-related symptoms by provoking this with total body radiation. If you give mice 11 grays of radiation, it's completely 100% lethal dose, but you can rescue them by syngeneic bone marrow transplantation. 100% of them survive, and we presume that they will be filled with senescent cells and start aging like crazy. The result appeared to be quite shocking. This is the trajectory of life of C57 black mice. If you irradiate them early in life and do bone marrow transplantation with a dose of radiation which makes every cell with damaged DNA, they live not too short a life. Only 20% life is only 20% shorter. But most amazingly is that when you measure frailty index, which is a cumulative parameter of the multiple physiological, biochemical, and other parameters, you will see that they are not worse than mice which were not irradiated. They're actually even a little better. So then we started looking where are senescent cells in these mice, which supposed to be there. And we looked at most famous biomarkers, which is P16. We have P16 luciferase mice, which indeed become positive for P16 luciferase late in life, as you see here. But mice which were radiated early in life, they don't have any acceleration of accumulation of P16. Moreover, they don't acquire any indications of inflammation, which would be faster than aged mice. So, and when you look at the gene expression profiling in major organs, you do not see any resemblance of transcriptome difference with aged mice, any resemblance to aged mice. And moreover, you do not even see indications of DNA damage response. This experiment we repeated many, many times, and we were quite shocked to see that. And of course, there was a big question: who are those cells which accumulate with age, which are P16 positive if they are not coming from direct DNA damage? That I'm not going to talk about. This is plenty of papers we published on that. It appears that P16 positive cells, which people believe are senescent in vivo, in reality are reprogrammed macrophages, which carry senescence-specific markers. And most importantly, these macrophages cannot be killed by senolytics, meaning that whatever we learned by killing, by rejuvenating of mice in which we killed P16 positive cells cannot be reproduced by senolytics because these cells are not senescent, they are macrophages. So you can read about that. I will move on with a more interesting story. So, being disappointed with senolytics, we started looking where are the cells with damaged DNA in these mice and why these mice don't age like crazy. So, and here we came up with the observation, which we reproduced already probably 100 times, and each of you welcome to do that. It's one of the most amazing phenomena I ever seen in my life of experimentalist. If you take these mice, which received 11 grays of radiation early in life, and if you take any organ of these mice, regardless, 1 week, 1 month, or 1 year post-irradiation, and you plate these organs as a single-cell suspension in vitro in culture, obviously in control mice, which were never irradiated, you see outgrowth of fibroblasts and monolayers in a week. Not a single cell would divide in mice which were irradiated, regardless how long ago you irradiated them. So, which means, and then we looked, and this, however, what is happening? It appears that 100% of their cells plated in culture, mesenchymal cells, they immediately activate DNA damage response, which they did not do when they were in vivo, and they activate senescent program and they acquire all the hallmarks of senescence. So then we looked what is happening with these cells in tissues, and it appears that all mesenchymal cells in these mice live with damaged, unrepaired DNA throughout the whole life of mice and only activate DNA damage repair if these cells force to divide. So this means that cells in highly irradiated animals, and probably humans as well, in mesenchymal tissue live under conditions of dormancy. So they are dormant, senescent-prone cells which turn into senescence only if they are forced to divide. So what is interesting that this is true only for mesenchymal cells. I can't say that there are no other tissues which would behave in the same way, but clearly not epithelial cells. Epithelium nicely repairs DNA and continue proliferating. So this phenomenon, DSPC, dormant senescent prone cells, is true for mesenchymal cells. So, of course, we were very interested to see. There is a mouse which looks like almost like normal mouse, but without a single mesenchymal cell capable of dividing. Is there any phenotype you can find in this mouse? Do you find conditions in which you can provoke cells to divide, and that should be converted into some kind of pathology? How to enforce it? Of course, the first idea which comes to mind is wound healing. Indeed, if you wound these mice, their wound healing is significantly impaired. They delay closure of the wounds, and most importantly, this closure of wounds happening without formation of a scar. It's only purely epithelial growth. So it's a phenotype, but not very impressive. much more impressive phenotype. We broke our brains trying to decide how to force mesenchymal cells to divide in adults when they're already adults. And it appears that there is a way. This way is to make mice obese. Because when you start putting mice into high-fat diet, you basically recruit tons of mesenchymal cells into adipocytic differentiation, the condition which requires single-cell division. So, and this is the result of these experiments. So this is these graphs you have already seen. If you give mice early in life high-fat diet without radiation, it's also aging-provoking treatment, and these mice also live shorter life, also about 20% shorter. Nothing impressive. However, if you combine radiation and then with high-fat diet, you see this result. So basically, obesity-provoking treatment leads to amazing acceleration of age-related symptoms and rapid death of these mice. Of course, one of the hypotheses was that if this is true and if obesity indeed leads to massive conversion of the cells into senescence because they are still, they want to divide, there was another prediction that if you do similar experiments without obesity though, but if you substitute obesity for being young and you take pups, which consist of cells which all dividing, it appears to be that the phenomenon is very nicely reproducible. You take 4-week-old mice, which are still growing like crazy. You give them bone marrow transplantation, but they don't live long life. They die very quickly. So most likely through the same mechanism as adults with obesity. So this brought us to a very interesting modification of the conventional view on what is happening following radiation in vivo. So this is what is textbook knowledge. You irradiate the animal, and depending on the cell types, you have either successful DNA repair, what we see in epithelial cells, or apoptosis, as we see in hematopoietic cells in the small intestine, or senescence, which leads to senescence-associated secretory phenotype and accelerated aging. Reality is different. The first two outcomes are the same, but most of the cells which are supposed to become senescent actually live with recorded damage in the form of broken DNA, but they never bother activate DNA damage response unless they're forced to divide in the form of dormant senescent prone cells, and only if you induce their proliferation you start seeing their bad influence, negative influence on longevity and health. So this paper is currently under review, in revision, and hopefully you will soon read it. It has tons of additional data I didn't have time to explain. So the bottom line for this first story, mesenchymal cells, in contrast to epithelial cells in tissues of adult animals, do not recognize DNA damage and do not activate senescent program and reside indefinitely as dormant senescent prone state, remembering that damage is there because they never bothered repair. Induction of adipogenesis, as well as any other way to induce their proliferation, leads to activation of senescence program and indication of accelerated aging. And this model most likely is the fastest model of accelerated aging I know of, in which you combine physiologically applicable and easily reproducible treatments. So, this is my first story, which actually adds interesting idea and additional interesting, peculiar modifications to our understanding what is happening with DNA damage in vivo. But now we're coming to the next story. The story is that this story does not contradict the idea that DNA damage has some relevance to aging. And if indeed DNA damage and accumulation of cells with damaged DNA contribute to aging, and aging is a genetically determined state, a process, then we need to have genetically determined mechanism of DNA damage. So the timer of longevity, if indeed as a driving mechanism it has accumulated the DNA damage, needs to be encoded in our genes. And I believe we know who that is. So here, I think this generator of DNA damage is residing in that half of our DNA, which consists of ancient virus-like elements, mostly represented by two classes of retrotransposons. Autonomous retrotransposons named line 1, encoding reverse transcriptase and producing reverse transcriptase to allow non-autonomous retrotransposons to also jump and create new copies named SINEs, short interspersed nuclear elements. Together they occupy nearly 40% of our DNA, and if you add to that other products of their side activity, such as processed pseudogenes, cDNA copies of mRNAs, you will get about 50%. So, which means that since all of these virus-like elements which appeared in our DNA through multiple explosions in evolution, there are millions of copies of them, they all replicate through the mechanism of reverse transcription. They are all retrotransposons or retroelements, and I think half of the genome deserves a term, so I introduced the term retrobiome, similar to microbiome, right? So half of our DNA I will name retrobiome. So this retrobiome is actually silent in normal cells, in vast majority of normal cells, under being under negative control of multiple mechanisms, including p53, Rb, histone deacetylases, and other. Well, as I said, they all replicate through the mechanism of reverse transcription, and the source of reverse transcription is line 1 elements. And the biggest gene family in our genes is actually, believe it or not, reverse transcriptase. We have 500,000 copies of line 1 sequences, 150 of which are still intact and technically can be activated if they're allowed to be genetically de-repressed and start generating new copies which will integrate randomly in any place of DNA, causing multiple unpleasant events such as mutagenesis, insertional mutagenesis provoking recombination, and this process, if it is activated, is recognized by the cell as viral invasion, and the cell activates interferon response as the mechanism of finding these cells and directing immune system towards them to kill them. So, for a long time, these events have been seen and considered as archaeological remains of our long past. I can tell you that now we are seeing that they are actually can be very effective contributors to our current pathologies. But before going there, I want to tell you something which people usually don't realize. So look at this. This is a schematic drawing of evolutionary tree, and most of mammalian species, orders of mammals, they bifurcated about 65 million years ago. And what is interesting that every order of mammals has its own SINE element. All primates, regardless with green monkey or human or chimpanzee or lemur, they all have ALU sequences. All predators have C1. All rodents have B1, B2, and so on. So what it means? It means that a real hypothesis was that what if this origin of species and acquisition of the way how we look somehow related to the invasions of line 1 sequences in our DNA. Indeed, if you look at paleontological evidence and look back about 165 million years ago, the time when dinosaurs were around, there were, if you would like at that time to make a zoo of mammals, this zoo would be very boring, because all mammals who lived at that time, according to paleontological evidence, looked pretty much the same. There was no that diversity of wonderfully looking creatures as we see now in the zoo. Then something happened. Dinosaurs extinct, and mammals became dominating animals on this planet. But what is interesting, look at this. All the major appearances, all the major morphological archetype of mammals appeared during very short evolutionary time, after which there was a long period, there was no big inventions in terms of forms. So after that, there was kind of Darwin evolution for adaptation to specific niches. I don't know, mouse and rat diverged, or hamster, dolphin and whale, and so on, but it's already happened within the archetypes, which appeared suddenly, shortly after dinosaurs died. What is very interesting, that exactly at that time, our genomes acquired SINE elements, and every order of mammals has its own class of SINE elements. This automatically leads to a very interesting reconstruction, that about 65 million years ago was a huge genetic pandemic when there was a big invasion or initiation of the process of massive acquisition of SINE elements in the predecessors of us. And this most likely led to the formation of lots of malforms and freaks, from which evolution picked those who managed to survive. And then, after those who survived, they initiated those orders which we see right now and enjoy in the zoo. This process most likely explains why we have such a huge amount of DNA with so little, so diluted, proportion of the the protein coding sequences. Because everybody at that time who had a compact genome didn't have chance to survive. Because when you have 200,000 insertional mutagenic events, if you have compact genome, each of them would be detrimental to your life and you would die. So, you may say that this all sounds like a fairy tale, and there could be many other explanations. And I can tell you no, because we have a proof that what I'm saying is true. And the proof is dogs. Domestic dogs there are multiple breeds. I'm sure that many of you have different breeds. And we all appreciate and take as granted that dogs are so diverse. But dogs represent the only known species which allows such a huge morphological diversity inside one species. There's not a single other domesticated animal in which humans were able to make such a huge diversity of forms. You may ask, what is so special in dogs? Why are they allowed to do that, and donkeys, camels, cows, horses, cats did not allow? And we know the answer. The answer simply was never summarized. But you can easily find this answer yourself if you simply read papers in which events which determine specific breed traits were identified. Today we know what is the mutation which created short legs of corgi or dachshund. We know what mutation created the flat nose of boxer and so on. Every time it's retrotransposon sitting in a new place. SINE elements, so which means that retrotransposons indeed, by expanding in DNA, create a huge number of morphological variations. They worked as morphogenes, from which, in this case, artificial selection allowed to pick up any form you like. So, this is, I hope you appreciate the remarkable role retrotransposon, when they get activated, they did with us, and they basically determine how we look. And obviously we live under conditions when this process has been stopped, and they are very significantly tamed, if you wish. Now we don't have active retrotransposon under normal conditions. But obviously that leads to a very interesting question. There are species which live exceptionally long life. Very well known naked mole rat, the species of rodent which lives 10 times at least longer than mouse or rat. So if indeed these retrotransposons had anything to do with aging, maybe if we compare the activity of this retrobiome in this species, we could find some hints. Why did I start thinking that they may be related to aging? For a very simple reason. Whenever you have in the cells activation of retrotransposon, this cell is prone to accumulation of DNA damage. And they are sitting in our DNA, and the degree of their negative regulation may be the determinant of speed of accumulation of cells with damaged DNA. And maybe by acquiring these SINE elements 65 million years ago, we acquired clocks of life, which is basically generator of DNA damage under control, and the depth of this control determines the longevity. So this is the hypothesis. So how can you look at the activity of a retrotransposon back in evolution? We don't have time machine, but we have we can do something else. And here I'm showing you the result of the work, which again is not unpublished but already written. And in this work, we managed to find a way how to measure activity of retrobiome back in evolution. I told you that side product of reverse transcription is a so-called processed pseudogenes. Sometimes reverse transcriptase, being promiscuous, makes copies of not its own mRNA, but mRNA of surrounding mRNAs, and the cDNA of surrounding mRNAs. After that, this mRNA cDNA copy goes back into the genome and creates completely inactive pseudogene because it doesn't have promoter. Promoter is not inside their coding region. So we can easily find pseudogenes. We have thousands of them in our DNA. And what we can measure the age of each pseudogen by the degree of its diversity from its own mRNA. The longer it sits in there, the more neutral mutation it accumulates because it's not under stabilizing selection, because it's not active. And you can easily look at the plethora array of all pseudogenes you can find and then classify them according to the degree of their diversity, how far they went from their ancestral mRNA. And by drawing this type of histogram, you can see how active was the process of retrobiome activity back to about 20 million years. That's the time we can find pseudogenes. So we have a proof that this idea really works. So this is the evolution of rodents, rats and mice divided into separate species about 10 million years ago, and naked mole rat, separated about 50 or 40 million years ago. So we first looked at the mouse and rat pseudogenes, and this is how it looks. You see that histograms of those pseudogenes which are unique for mouse and rat, they are at the left and right. And in the middle are pseudogenes which are common between them, those which appeared in evolution at the time of their common predecessor. As you see, in these common pseudogenes, there are none which would have 100% homology to the mRNA because they live at their ages at least 10 million years. At the same time, you see that there are plenty of pseudogenes which are very close to their ancestral mRNA in species which, among those which are recently acquired. But then, when what attracted us here is the following. It looks like in mice, there are plenty of pseudogenes which are identical in sequence to mRNAs. This means that this process in mice is going on. To test that, we took 12 C57 black mice from Charles River, and we sequenced them. And we analyzed pseudogen contents. And we found diversity of pseudogenes among these Charles River mice, which is supposed to be completely inbred. So this is a proof that this process is indeed ongoing. But this is the most shocking result. Look at the histogram of which basically represents the relative activity of retrobiome during evolution from now back to 15 million years ago in mouse and naked mole rat. You see that the profiles of this histogram is completely different, meaning that about 10 million years ago, the precursors of naked mole rats passed through the period when the retrobiome was extremely active. They created tons of pseudogenes, many more than in mice. But then this process was completely stopped. And they have no pseudogenes, which would be identical to the mRNA, indicating that they today completely suppress the process of retrobiome activity. And this is the reconstruction what happened. So many years ago, the common ancestor of rodents lived, and suddenly in some subfamilies there was a massive explosion of SINE elements, activity of retrobiome, which most likely led to massive extinction and malformation, and those who happened to survive somehow managed to stop this process. And together with that, they acquired much longer longevity. It's not a proof that there is a connection, but it's a nice illustration that connection could be. So, and this is, by the way, comparison of different species of animals, including humans, for the proportion of pseudogenes which are identical, recent pseudogenes. You see that naked mole rat is a big outlier here. So, all this together brought us to this idea that together with acquisition of SINE elements, we acquired the clock of life. This clock of life is in the form of retrobiome, which with certain frequency spontaneously happen to overcome the negative regulation and start making cells with damaged DNA, which determines the differential lifespan in different species. So, if this is true, then aging potentially, at least the aging in terms of accumulation of damaged DNA, can be considered as a viral disease, and as such can be treated as viral disease. And fortunately for us, we can do it. How we can do it? We can do it because reverse transcriptase has been a nice target for treatment of HIV and hepatitis B. And we have tons of drugs which are, on one hand, very efficacious inhibitors of reverse transcriptase, and on the other hand, they are promiscuous enough, non-selective enough to be effective against reverse transcriptase of line 1. So this is papers, some of which are from us, some of which are not, which demonstrate that activation of retrobiome indeed induces not only DNA damage but also inflammation. I already mentioned that it activates interferon, and through C-gas sting pathway activates an F-kappa B as well, and that this process is druggable. So, which means that you can use reverse transcriptase inhibitors and stop this process. Of course, and this brings us back to the beginning of my talk when I said that if there is a clock of life, it should be genetically determined, should be associated with accumulation of damaged and somatic cells, and leads to systemic inflammation. And all these things is being done by retrobiome, meaning that making it a interesting candidate for this role. So, of course, when you have a drug which you would like to apply to control retrobiome, you cannot start from aging. You need to introduce it against some other process which can be more effectively measured and easier to supported by funding. And retrobiome activity plays a big role in cancer progression. So now we know that a big proportion of p53 function, and p53 acts, as you know, as a stabilizer of genome and epigenome. It happens because p53 is a repressor of retrobiome. And when you use inhibitors of reverse transcriptase, they act as partial revertants of p53 activity in tumors where this activity is lost by inhibition of inflammation associated with retrotranspositions of line 1 elements. So what is in the paper which we just published several months ago, we demonstrated that activation of line 1 elements in tumors, which frequently happens in human tumors, is a big driver of cancer resistant to treatment, and it can be reverted by reverse transcriptase inhibitors, which by themselves do not have any anti-cancer activity, but they block cancer ability to adapt. So all this, of course, opens up the possibility of using this approach to control retrobiome and aging, and these experiments we obviously started from our our favorite model of accelerated aging when DNA damage with radiation combined with obesity. And these are two curves, mice which were just irradiated and mice which were irradiated, lived a normal life, and then were put on high-fat diet and start dying much faster. But then you take another third group of mice which were put on high-fat diet together with reverse transcriptase inhibitor and drinking water. And these mice live substantially better, indicating that we are most likely on the right track. So I'm wrapping up, saying just to give you several messages which I tried to deliver, that repeats, which used to be named repeatome, I prefer the term retrobiome, which occupies nearly half of our DNA. It determines the way we look. It played a big role of morphogene in our evolution. It is likely contributed to mammalian aging, and it definitely contributed to cancer progression and treatment resistance. And it is a target for treatment of both diseases. Everything looks very rosy and great, but I would like to finish by making a kind of negative, disappointing point. If we have in our genes something for a couple of dozens of millions of years, almost unavoidably it needs to be useful for something. And indeed, it appears that the same process which I explained to you, activation of inflammation in the cells which happen to allow retrobiome to be active, plays a very important role in cancer prevention, because it directs immune system to the cells which happen to lose control over their epigenome. And therefore, using inhibitors of reverse transcriptase in normal people as a matter of prophylaxis, I would not recommend. Although initially I thought that this Lucas Cranach famous picture finally can be interpreted as having reverse transcriptase inhibitors in this pool. Therefore, retrobiome is an interesting illustration to the story of Dr. Jekyll and Mr. Hyde, being on one hand bad guy by inducing mutagenesis, accelerating aging, and cancer progression, but at the same time it's a major cancer preventive agent because it allows immune system to find and kill cells which are decided to step on the way of crime. So where we're going to be eventually and how to use reverse transcriptase inhibitors, I don't know, but one thing is clear to me. They most likely will be used with cancer treatment because they definitely can block cancer progression. I will finish here. Thank you.